Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Did you know that your brain contains not just like one type of nerve cell, but hundreds, maybe even thousands?
Yeah, and each one's unique, right?
Yeah.
Different job, different look, different electrical buzz.
Exactly. And for so long, scientists could only grow maybe a few dozen types in the lab.
Which sounds like a lot, but it's really not. When you think about the sheer complexity of the brain, it's like trying to understand a whole city by just looking at one street corner.
Right. And that limitation, well, it's been a huge roadblock for neuroscience, hasn't it?
Oh, absolutely. For decades.
Right.
If you're trying to figure out diseases, you really need to isolate the specific cells involved.
You need the right pieces of the puzzle.
Precisely. Without that, it's incredibly difficult.
Okay, so that brings us to today. We're doing a deep dive into some really groundbreaking research from ETH Zurich.
Ah, yes, the work on generating neurons.
Exactly. They've massively expanded our ability to create human nerve cells in the lab. We're talking over 400 distinct types.
400.
Yeah,
That's a huge leap. It's not just a small step forward.
Right. So for this dive, we're looking at a detailed paper from Science and also a piece from Science Daily that covers the work.
Good sources. The science paper really gets into the nuts and bolts, the methodology.
Yeah, and Science Daily helps put it in context. So our mission today is basically to unpack how they did it, what this means for treating really tough neurological diseases, think Alzheimer's, Parkinson's, and what this unlocks for personalized medicine down the line.
Sounds like a plan. Where do we start? The challenge itself.
Let's do that. So for ages, the big problem, as we said, was growing a real variety of human nerve cells in the lab.
Right. You'd either get these messy mixtures of different cells or maybe just a handful of specific types. Not ideal.
Not at all. You can't really study something like, say, epilepsy effectively if you can't get your hands on the specific neurons that go haywire during a seizure.
So how did they tackle this? What was the core idea?
Well, they focused on something called induced neurons. Let's call them INs for now, just easier.
Okay, induced neurons. So these are human neurons made in the lab.
Yeah, both excitatory and inhibitory ones. They're generated from pluripotent stem cells.
Ah, the stem cells, the ones that can become almost anything, like a blank slate.
Exactly. Super important for studying how nerves develop and what goes wrong in disease. But getting them to turn into specific types of neurons, that was the tricky part.
Right. So how do you tell a blank slate cell, okay, I want you to become this specific type of forebrain neuron?
This is where it gets clever. They use things called pioneer transcription factors.
Okay, transcription factors. Those are proteins that control which genes are turned on or off, right?
Precisely. And these pioneer ones are like master keys. They can unlock specific genetic programs to push a cell towards becoming a neuron.
And they use specific ones in the study.
They did. They focused on neurogenin 2 and also aceta-skewed homolog 1 as key examples. Powerful stuff.
Okay, so you've got the stem cells and the factors to push them towards being neurons. What else?
Then they brought in morphogens.
Morphogens. Those are the signaling molecules from embryonic development.
You got it. Think of them as like chemical messengers or navigators. In an embryo, they tell cells where to go and what identity to take on.
So they help establish different brain regions, for example, like forebrain versus midbrain.
Exactly that. They provide the positional information.
Okay, so pioneer factors for the become a neuron signal, and morphogens for the become this specific kind of neuron signal. Makes sense.
And here's the really innovative part. They didn't just guess which morphogens to use. They set up this huge systematic screening process.
Systematic how?
They tested 480 unique combinations. Different morphogens, different concentrations, all combined with those pioneer factors.
Wow, 480. That's a massive experiment. Just managing that must have been complex.
Totally. And to figure out what actually happened in each of those conditions, they used a technique called high-throughput single-cell ribonucleic acid sequencing.
Ah, single-cell RNA-seq, looking at the gene activity in each individual cell.
Exactly. They profiled nearly 700,000 individual cells.
700,000. The amount of data must be staggering.
It gives you this incredibly detailed picture of how each cell responded, which genes switched on or off under each specific morphogen cocktail.
Okay, so they did this massive screen, generated tons of data. What did they actually find? What kinds of neurons could they make?
Well, the big headline is they successfully created this huge diversity of induced neuron subtypes. And crucially, these lab-grown cells closely resembled the ones found naturally in the human body.
So a much bigger toolkit for researchers now.
A much, much bigger toolkit. But you asked earlier how close they are to the real thing.
Yeah, is it just sort of similar, or are they really capturing the key features?
They found remarkable specificity. They could generate neurons that match those from distinct regions: forebrain, midbrain, hindbrain, even spinal cord, and the peripheral nervous system.
That's huge. So you could, in theory, generate midbrain dopamine neurons relevant to Parkinson's.
That's the idea. Or specific spinal cord neurons for studying motor neuron disease. The potential is enormous.
And functionally, did they behave like real neurons?
Yes. They produced the right kinds of chemical messengers.
Yeah.
Neurotransmitters like glutamate, GABA, dopamine, acetylcholine.
The key signaling molecules in the brain.
Right. And their electrical activity patterns were diverse and matched what you'd expect from different neuron types. They weren't just looking the part, they were acting the part too.
Okay, so they achieved this diversity. How did they figure out the mechanism? The how?
They dug into the gene activity data. They mapped out these things called gene regulatory networks.
Right, the complex web of genes controlling each other inside the cell.
Exactly. And within those, they looked at regulins. A regulon is basically one of those master switch transcription factors and all the specific genes it directly controls.
So they could see which morphogens were activating which specific networks or regulons to create a particular neuron type.
Precisely. They could trace the pathway from the external morphogen signal to the internal genetic program being activated.
And they tested this. They didn't just rely on the correlations in the data.
Oh yeah, they validated it. They did experiments where they removed key transcription factors identified by their analysis.
Yeah.
And sure enough, without that factor, the morphogens couldn't push the cells into that specific subtype anymore.
Okay, that's strong evidence.
And they did the reverse too. They took cells and just overexpressed one of those key factors.
What happened then?
It was enough to drive the cells towards that specific subtype, even without the morphogen signal.
Wow. So that really pins down the role of those specific factors in determining the cell's fate.
It really does. It shows a real level of control over the programming.
That's fascinating. Anything else that stood out about the how?
Yes, something really interesting about timing.
Timing.
Yeah, when they exposed the cells to the morphogens, made a big difference. They tried two main approaches.
Okay.
One was pre-patterning. That's where they treated the pluripotent stem cells with the morphogens first, before they even turned on the pioneer transcription factors to make them neurons.
Ah, so sort of setting the regional identity before the neural identity.
Exactly. And they found this actually yielded neuron subtypes that were more uniform, more like primary human neurons in some ways.
Interesting. Any downsides to that approach?
Well, initially, these pre-patterned cells were a bit less mature, and there were sometimes more off-target cells that didn't quite become the intended neuron type.
Okay, so a trade-off. And the other approach?
That was post-patterning, adding the morphogens after turning on the pioneer factors.
So making them neurons first, then telling them what kind?
Right. And the results were different. It just highlights how sensitive this whole process is. It's not just what signals you give the cells, but when you give them.
Nuance matters. Okay, so let's pivot a bit. We've talked about the how. What are the big implications here? What does this mean for, medicine and understanding the brain?
I mean, it's potentially revolutionary for studying neurological diseases. One expert mentioned in the coverage, Barbara Treutlein, really emphasized this.
What was her point?
That for so many diseases, Alzheimer's, Parkinson's, epilepsy, even things like sleep disorders or multiple sclerosis, researchers desperately need to study the specific nerve cell type involved.
Right, because it's usually not the whole brain that's affected equally. It's specific populations of cells.
Exactly.
Yeah.
And now, with this ability to generate hundreds of specific types in the lab, well, you can finally create much more accurate models of these diseases.
You can build the disease in a dish using the right cellular building blocks.
Pretty much. Imagine having the precise cellular Lego bricks. You need to really see what's going wrong cell by cell.
That has to speed things up.
Hugely. Especially for drug discovery.
How so?
Well, think about it. Right now, a lot of drug testing relies on animal models, which aren't always perfect translations to humans, or on less specific cell cultures.
True.
Now, you can grow the actual human nerve cells relevant to Parkinson's, for example. Then you can test potential drugs directly on those cells in a dish.
So faster screening, more relevant results, hopefully leading to treatments getting to patients quicker.
That's the hope. And it could potentially reduce reliance on animal testing, too.
That's a significant side benefit. What about therapies themselves? Does this help there?
Potentially, yes.
Yeah.
It brings things like cell replacement therapies a step closer.
You mean actually replacing damaged or dead neurons in the brain with new healthy ones?
Exactly. If you can reliably grow specific, functional human neurons in the lab, that's a critical first step towards being able to transplant them therapeutically. Still a long way off, probably, but this is foundational work.
Definitely laying the groundwork.
And another key point the researchers showed, this whole systematic approach isn't just for neurons.
Oh.
Yeah, they demonstrated it's generalizable. It worked with different pioneer factors, and the basic principle, systematically testing morphogens or other signaling molecules, could likely be used to program other kinds of cells too.
So this could have implications beyond neuroscience for regenerative medicine more broadly.
Potentially, yes. It opens up a lot of doors for understanding how to control cell fate in general.
Okay, this all sounds incredibly promising, but science always has nuances, limitations. What are the caveats here?
Good point. It's important to be realistic. While they massively improved the diversity and the purity, getting up to like 80% homogeneity for some specific types, most cultures still weren't perfectly pure.
So you still get a mix, just maybe a much better defined mix or a higher percentage of the desired cell?
Right. Getting to completely homogeneous cultures of one specific type, that likely needs more work. Fine-tuning the timing, maybe adding other factors.
Still a challenge to get 100% purity. What else?
There are also some differences identified between these lab-grown induced neurons and actual primary neurons from the human body.
Ah, okay. Even though they looked and acted similarly in many ways.
Yeah. In terms of neurotransmitters, ion channels, gene expression patterns, they were quite similar. But the study noted significant differences in their metabolism.
Metabolism, like how they produce energy.
Exactly. Things like oxidative phosphorylation. The lab-grown cells weren't quite identical metabolically.
And that could matter for how they function long term or how they respond to certain drugs or stresses.
Potentially, yes. So figuring out how to correct those metabolic differences, make them even more like native neurons, that's a key area for future research.
Got it. Any other limitations?
There's also the issue of those off-target cells we mentioned earlier.
Right, the ones that didn't quite become the intended neuron type.
Yeah. In some conditions, especially using certain combinations of pioneer factors like a Chady-skewed homolog 1 with another factor called DLX2, they got a higher proportion of these non-neuronal cells.
Suggesting that maybe not all pioneer factors work equally well on their own, or they might need specific partners to be really efficient.
Something like that.
Yeah.
It's another piece of the puzzle they need to solve to make the process even more efficient and reliable across the board.
Okay, so still work to be done, but a massive leap forward nonetheless.
Absolutely. So wrapping this up, what's the big takeaway for you listening to this?
Well, I think it shows the power of combining deep knowledge of developmental biology.
Yeah.
You know, how embryos build brains with really systematic, large-scale experimentation.
Yeah, it wasn't just a lucky guess. It was methodical.
Right. And it's moving us from this kind of general understanding of nerve cells towards this incredibly precise ability to make and study the specific cells that matter for health and disease.
We're getting much more granular, much more specific.
Exactly. And this research, it really feels like a huge step towards that future people talk about, where we can accurately model brain diseases in a dish, maybe even patient-specific diseases.
Pinpoint the exact cell type causing the problem.
And then maybe even engineer healthy replacements. It enables targeting specific neuron types for modeling, for drug testing, for potential therapies. It's really about precision.
A move towards precision medicine for the brain. So here's something to think about. With this new power to engineer specific brain cells, which neurological diseases do you think stand to benefit the most, the fastest, from this kind of targeted approach?
That's a great question to ponder. The possibilities seem vast.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.